Okay, so ladies and gentlemen, you are all welcome to my YouTube channel. Today we are going to look at the qualitative test for amino acids. Qualitative test for amino acid, but we need to understand what is amino acids. So what is amino acid? Amino acid is a building block of proteins. It means that proteins are what actually make, or the proteins are made up of an amino acids. So it's like in a proteins, there are blocks that are used in building proteins, and these blocks that are used in building the proteins are called amino acids. And the structure of this amino acid, it consists of two major functional groups. It consists of two major functional groups, and these two major functional groups it consists of an amino group. It consists of an amino group, carboxylic group, and there is also an R side chain. And of course, this is the general structure of an amino acid. This is the amino group, this is the carboxylic group, and this is the R side chain. And of course, there is a free hydrogen. And this R side chain is actually what make one amino acid differ from one another. Like in glycogen, the R side chain is hydrogen. Like in alanine, the R side chain is CH3. So each of the amino acid is specified by its own R side group. And of course, currently there are about more than 300 amino acids that are known. There are more than 300 amino acids that are known, but only 20 are found in proteins. So there are 20 naturally occurring amino acids that are found in proteins. So the 20 naturally occurring amino acids are the essential amino acids, which are broadly classified into three types. They are broadly classified into different groups, and this group could include polar amino acids. So when we say polar amino acid, it means that they are the amino acids that possess a functional group like COOH and amino group, and these functional groups make these amino acids to form hydrogen bond with water, and as a result of that, they can interact with water. So polar, it means that they like to interact with water. And then there is also some amino acids that are called non-polar, and they are non-polar because they possess aliphatic or aromatic R side chains and hydrophilic in nature. So it means that this kind of non-polar amino acid are those amino acids that, of course, this is a mistake. It's supposed to be hydrophobic in nature. It's supposed to be hydrophobic, not hydrophilic. So they are hydrophobic in nature. They are hydrophobic in nature because of the presence of aliphatic and aromatic side chains. And then the next thing, there is also acidic amino acid, and this acidic amino acids are those amino acids that comprise of carboxylic acid on their side chains, and we have basic amino acids, and those basic amino acids are those that have basic side chain. So this is amino acid, and we have also another classification of amino acid. We have them based on the nutrition, which is essential and non-essential amino acids. So we said essential amino acids are those amino acids that cannot be synthesized by the body. So we have essential and non-essential. So we have classification of amino acid on different bases, but in these cases, we are trying to look at some basic explanation of amino acids. So as I said, the focus of today's class is actually a test. It is qualitative test for amino acids. Qualitative test for amino acids. We are going to look at qualitative test for amino acids. So we said qualitative test from the word qualitative test for amino acids. Qualitative test for amino acids. So when we said qualitative test, it means to check the presence or absence of amino acids in a given So qualitative test, it means to check the presence or absence of the amino acid. So you'll be given a solution. So when you are given a solution, to test the presence or absence of an amino acid in a given solution is what we call qualitative test. So generally in chemistry or in biochemistry, When we say qualitative test, it means that is check the presence or absence of a particular analyte. So that is the qualitative test. And for the quantitative test is to check the amount or the concentration of the given analyte. Like for example, if you now have, let's say you have your test tube, and in the test tube you have a solution. So inside this solution, maybe you want to detect the presence of an amino acid. Let's say lysine. You want to detect the presence of lysine. So to check the presence or absence of lysine in this given particular solution inside this test tube, to check the absence or presence of this lysine is what we call qualitative test. But for the quantitative test, you already have an idea that, okay, in the solution you have lysine. But to know how much of that lysine is present in the given solution is what you call quantitative. Quantitative, that is to quantify the amount of the lysine in the given solution. So that is quantitative. So now we are starting with this qualitative test. So for the qualitative test, the first one that we are going to look at is a non-hydrogen test. We are starting with non-hydrogen tests. Non-hydrogen test. So what is non-hydrogen test? It's a test that is used for all given amino acids. So if you want to check the presence of absence of any amino acid, so you apply or you use a non-hydrogen test. Assuming that you are given a solution and you want to know whether in the solution you have a particular amino acid, because non-hydrogen test is not specific. It's a test that is used to test any types of amino acid. You can use non-hydrogen test to check the presence of the amino acid or absent any of the amino acid, any class of the amino acid. As we said that we have 20 amino acids, so to check the presence or absence of 20 amino acids, you can apply or you can use non-hydrogen test. So this test is actually due to the reaction between the amino group of an amino acid and the non-hydrogen. So the reaction between the amino group and the non-hydrogen is actually the basic principle behind the non-hydrogen test. And this non-hydrogen is a powerful oxidizing agent, and it is present. So it is a powerful oxidizing agent, and it is present amino, and it is present. So amino acids undergo the presence of this non-hydrogen in a given solution. So it will actually oxidize amino acids through what we call oxidative deamination. So this oxidative deamination by this non-hydrogen leads to the release of ammonia, carbon dioxide, and the corresponding aldehyde. So it means that when non-hydrogen reacts with amino acids, it liberates ammonia, carbon dioxide, and non-hydrogen. So it means that you have amino acid. When you have your amino acid plus non-hydrogen, then you are going to have ammonia in history. You are going to have plus carbon dioxide, then plus also aldehydes. So this is what actually happens if you now react it amino acid with non-hydrogen, then we are going to get this. So after we get this, the non-hydrogen itself will now reduce to produce what we call hydrogen dantin. And this non-hydrogen dantin also further, like for example, the amino group formed from the amino acid, it will now react with another molecule of non-hydrogen and is now reduced product hydrogen dantin to form a blue substance called Rohemann's complex. So what does that mean? It means that first, when amino acids react with non-hydrogen, it produces ammonia, and this ammonia is actually form part of the non-hydrogen. which actually form a complex called ninhydrindanthene. And it's this ninhydrindanthene which actually contain this ammonia, then react with another molecules of ninhydrin. And once it reacts with it, then it now produces a blue substance or a blue complex known as Rohemann's complex. So however, in the case of amino acid like proline and hydroxyproline, a different product having a bright yellow color is formed. So it means that in this situation where we have when a proline and the hydroxyproline react with the ninhydrin, then it forms a yellow color, a bright yellow color. But in the case of asparagine, because it has a free amide group, then it will react to produce a brown colored product. So now let's actually look at the reactions really. So this is actually like how the color is produced. So this is a purple color. That is, this actually detect that in the detection, it means that yes, there is amino acid when you observe a purple color. That is for general all other acids, except for proline, hydroxy, proline, and asparagine. So this is the reaction. First, you have ninhydrin react with amino acid to form an intermediate amine called hydrindanthene, aldehydes, and then carbon dioxide. So this intermediate amine will now react with another ninhydrin, then to form a Rohemann's purple color complex. So whenever you observe the purple color, it means that the amino acid is present in the given solution. So that is ninhydrin test. So the principle is behind the ninhydrin react with amino acid to produce intermediates called hydrindanthene, and this hydrindanthene now react with another ninhydrin to form Rohemann's purple color complex. So these are the materials needed. You need test solution, and in the test solution we expect to have 1% alanine, 1% proline, and 1% asparagine. And you also have 2% ninhydrin in an acetone. So we'll add 2% of ninhydrin in an acetone. Then you have water bath, dry test tube, and pipette. These are the material that you are needed. So the preparation of the 2% ninhydrin, what you are going to do is actually you are going to weigh 0.2 gram of ninhydrin, and then you dissolve it in 10 ml of acetone or ethanol. Then the preparation of the test solution. So if you prepare 1% amino acid solution, then you add it in a distilled water. So after you have that, you now take 1 ml of the test solution and you put it in a dry test tube, and you also take another 1 ml of distilled water, you add it into another test tube, which can serve as a control. Then you now add few drops of 2% ninhydrin in both the test tubes, the test tube that contains your amino acid solution and that of the control. And then you keep the test tube in water bath for 15 minutes, and then you look for the development of blue or violet color, and you observe that. But in the case of proline and hydroxyproline, yellow color will be developed instead of blue color or purple color. Similarly, also in the case of asparagine, it will give a brown color. So then the next thing is hypoxanthine. So xanthoproitic test. Next one is xanthoproitic test. So what is xanthoproitic test? So the next thing is xanthoproitic test. So this test is specific for detecting aromatic amino acids. So we have 3 major aromatic amino acids, and these 3 basic aromatic amino acids we have. One, we have phenylalanine, we have phenylalanine, we have tyrosine, we have tyrosine, and then we have tryptophan. So these are the aromatic amino acids. So this xanthoproitic test is actually specific for the aromatic amino acid, that is those amino acids that contain benzene ring or benzene nucleus. The xanthoproitic test is actually based on the principles of nitrification reaction. In this test, the active aromatic amino acid undergo nitrification in the presence of concentrated nitric acid and form a yellow colored nitro derivatives. So it means that if you have aromatic amino acid, then the amino groups, then the aromatic amino acid, it will now undergo nitrification, that is addition of nitrogen. And this is actually happening in the presence of nitric acids. So for example, let's see, this is your aromatic amino acid, aromatic amino acid. So in the presence of concentrated nitric acids, okay, it will now form a nitro color derivatives. It will now form a color yellow. It will now form a yellow colored nitro derivative. So it will now form a color complex. So, and of course, the yellow color turns into orange by ionization of the phenolics group at an alkaline pH. So the method: first, you prepare 1 ml of solution of a given sample, then you now add a few drops of the nitric acid, and then you boil it in the water bath for 2 minutes, and then you allow it to cool, and then you observe the color. So the appearance of yellow color indicates the presence of aromatic amino acid. And of course, this is a color here, where the yellow color here indicates the presence of an aromatic amino acid such as tyrosine and tryptophan. So it means that this type of test is specific for aromatic amino acids. So the next thing we have also Foley's test. So in a Foley's test is a test that is specific for amino acid like histidine and tyrosine. So it's a specific test that used in detection of histidine and tyrosine. So the principle of Foley's test is based on diazo tests. And this diazo tests, the principle of Foley's diazo test is based on the diazotization reaction. So what is the diazotization reaction? Generally, first we need to understand that the Foley's reagent is made up of some components, and this component include sulfonilic acids. And this sulfonilic acid is the one that undergo diazotization reactions to form a diazonium salt in the presence of sodium nitrate and hydrochloric acids. So now, for example, first look at this is the reaction. We have sulfonilic acid which react with nitric acid plus HCl to produce diazonium salt plus water. And this diazonium salt react with tyrosine to produce azodine, which is actually red in color. And the appearance of the red color in the amino acid using the Foley's diazo reagent, it means that you have histidine and tyrosine. So red color indicates the presence of histidine and tyrosine in the Foley's diazo tests. So these are the requirements. So you need the reagent, you need 1% sulfonilic acid, and you chill it. And you need 10% hydrochloric acid, 5% sodium nitrates. You also chill it, and you need 10% sodium carbonate. And then you now chill the samples containing the tyrosine and histidine. And these are the materials: iceberg, vortex, test tube, pipette, and test tube stand. So the procedure: first, a test tube, then you add 1 ml of chilled sulfonilic acids, and then you add a few drops of free chilled sodium nitrate and mix in a vortex. So you vortex it, then you now add 1 ml of the amino acid sample. It will be immediately added to the test tube, and then you mix it. Then you add few drops of sodium carbonate, and then you add to the test tube drop by drop until color begins to appear. So the presence of red color complex indicates the presence of the tyrosine and histidine, while the absence of the red color indicates that histidine and tyrosine is absent in these types of tests, which is called Foley's test. So now the next thing is Sakaguchi test. So Sakaguchi test is a biochemical test consisting of colorimetric reaction and is usually used for detection and quantification of amino acid with guanidinium groups, which is actually as a qualitative test for arginine that is either free or in a protein. So arginine is an amino acid that contains guanidinium groups. So you can use Sakaguchi's test to actually either to test it using a qualitative method. So this Sakaguchi's test is an example of color reaction or a test that is performed for the detection of amino acid or protein. So if you are given a samples, you can ask from the samples to detect the presence of either the free amino acid or any protein that contains the arginine amino acids. So the test is specific test for arginine where the guanidine groups of the arginine will react with alpha naphtol or 1-naphtol to produce a color product. So it is a qualitative test, and the quantification of arginine is hindered in this test due to the slow rate of color development and the destruction of some guanidinium groups by the reagent. So it means that there is a limitation. So in this test, the first thing that we need to know is in Sakaguchi test, is a test that is specific for testing the presence of arginine, that is amino acid that contain guanidinium group. And it is the guanidinium groups of the amino acid that react with either the alpha naphtol or 1-naphtol to produce a color product. And this test and the quantification can be hindered. There is a limitation due to the slow rate of color development or the destruction of the guanidinium groups by the reagent. That is like, for example, there are some reagents like hypochlorites. If you have hypochlorite, it can destroy the guanidinium group. So if it destroys it, it means that it may give a false positive. But also the slow reactions, the slow color formation can also affect tests. And the main objectives of Sakaguchi's test is to detect the presence of arginine in a free form or in a protein. So you can detect the arginine in both, either as a free amino acid or in proteins. So what are the principles of Sakaguchi's test? The principle of Sakaguchi's test is based actually on the principles of reaction between the alpha naphtol or 1-naphtol and the guanidinium groups in the arginine in the presence of the oxidizing agent, and the exact mechanisms of the reaction is not yet known. However, the reaction results in the formation of red-colored complex with the formation of indole-like structure. So the Sakaguchi's reaction, it consists of hypobromides and 1-naphtol. And of course, the sodium hypobromide act as an oxidizing agent that facilitates the hydrogen bonding between the two arginines. So this is, you have the arginine and you have alpha naphtol. So two molecules of alpha naphtol react with a guanidinium group of the arginine in the presence of sodium bromide, water, sodium bromide, and hydrogen peroxide. Then it will now form a red colored complex. These are the requirements. In Sakaguchi's test is a reagent that contains 1% of 1-naphtol in alcohol with a few drops of 10% hypobromide solution of amine water. And there is also 40% of sodium hydroxides and also the sample. So these are the reagents that I require, and the materials. We need test tubes, we need test tube stand and pipette. For the procedure of Sakaguchi's test, about 3 ml of the test solution is added in a test tube, to which 1 ml of 40% sodium hydroxide is added and mixed correctly. Then 2 ml of 1-naphtol are added to the same sample test and then mixed thoroughly. Then now you add 4 to 5 drops of the 10% sodium hypobromide or bromine water is added. Then the test is observed for the development of color. So for the negative test, if there is no arginine, it means that it will give us the red color complex will be absent, and if there is the presence of guanidinium group in the arginine, there won't be a red color formation. So these are the limitations of the test. So qualitative test for the colored product is not possible with the test as the rate of several which reaction is very low. And similarly, some of the quantitative groups in the solution might be destroyed by hypochlorites, resulting in difficulties in testing the results. So the next thing, which is Emilian's test. Emilian's test is also an analytical test used for detection of amino acid tyrosine, which is only amino acid containing phenol group. So it means that Emilian's test is used for testing amino acid that contain phenol groups. And this Emilian's test is a specific test for tyrosine, but it's not a specific test for protein, as it also detects the phenolic groups present in other compounds as well. So it means that this is a limitation, because since apart from the tyrosine, if there is any amino acids or if there is any compound that contains phenol groups, it means that it will detect it. So therefore, while we are calling Emilian's test, it is essential that other tests like Barrett's test and then hydrogen tests also be performed, so that to confirm that what you are detecting is really the tyrosine. So as many proteins consist of tyrosine, the test is useful in the detection of such proteins, and the test was discovered by and named after the French chemist Auguste Nicolas Eugène Milan. And the objectives of the Milan test first is to detect the presence of tyrosine containing proteins in a given sample. It is also to detect the presence of phenol containing compound. And then lastly, to differentiate tyrosine from other amino acids. So what are the principles of Milan's test? The Milan's test is based on the principle of nitrification of phenolic group in tyrosine. So it's through a nitrification reaction, which then forms a complex with a heavy metal like mercury. So the reagent used for the test is called Milan's test, and it consists of mercuric nitrate and mercurous nitrate, which is dissolved in concentrated nitric acid. So it means that the component of Milan's reagent consists of mercuric nitrates. It also consists of mercurous nitrate that is dissolved in nitrate. So it means that there are three components: mercuric nitrate, mercurous nitrate, and then the nitric acid. So in the test, the phenolic group on the tyrosine molecules is nitrated by nitric acid present in the reagent, and then the nitrated tyrosine then combined with the mercury ions in the solutions to form a red colored precipitate or solution. Or in some proteins containing tyrosine, the initial reaction between the mercuric nitrate results in a white or yellow colored precipitate. And after the addition of nitric acid and heating, however, the residue turns red in color, and both of these results are considered positive results and indicates the presence of tyrosine in a solution. So these are the reagents and the materials that are needed in this test. We have the Milan's reagent, and then preparation of the Milan's reagent, and then the materials. So these are, because in the Milan's test, these are the first. Either you add 2 ml of a sample solution, or the 1% tyrosine solution is taken into a test tube. And to this, about 2 ml of Milan's reagent is added, and then the test tube are then kept in water bath for about 2 minutes if red colored precipitate is not observed immediately. And then the test tube are then observed for the formation of the colored precipitates. So the red color indicates the presence of tyrosine or phenol containing compounds. And absence of the red color indicates that, or the absence of red or pink color indicates the absence of the amino acids. So ladies and gentlemen, thank you for attending, or thank you for listening. And if it is the first time you are visiting my YouTube channel, please subscribe.